In this video, we will study about gene expression. Now, what is gene expression? What is its mechanism? What is central dogma? What is central dogma reverse? What is one-way flow of genetic information? Circular flow of genetic information and reverse transcription. Now we'll discuss what is gene expression. Now, it is the mechanism at the molecular level by which a gene is able to express itself in the phenotype of an organism. Now, what is phenotype? This is the morphological character, external appearance. If we talk about height of the plant, so this is being determined by a gene, whether the plant is tall, whether the plant is dwarf, or what is the color of the flower, again that will be determined by the gene. So whether the color is purple, pink or white, so many other characters, they are determined by the gene. So they control the phenotype of an organism, but this work is done at the biochemical level. This is called as biochemical genetics. That is, it leads to synthesis of RNAs, enzymes, structural proteins and polypeptide chains. During this process, what happens? A part of the DNA we know is deoxyribonucleic acid, will act like a gene. This is called as cistron. Now, DNA has bases, nitrogenous bases, adenine, thymine, guanine, cytosine. They show complementary pairing with the other strand, like opposite A, T, opposite T, A, opposite G, C. For learning about nucleotides, you can watch my video of nucleotides. Link is shared in the description box. Now, DNA has codons, and those codons, then they are transferred, that message is transferred in the form of RNA, and this conversion of one strand of DNA to RNA, strand of the DNA which act as complementary to RNA, that is called as template strand, and messages on the DNA, they are carried to the RNA. This is called as transcription process. Again, to learn about transcription, you can see my video transcription 1 and transcription 2. Link is shared in the description box. Now RNA will be converted to protein. This is called as translation. First we learn DNA can replicate, mean it's two strands, they can get separated and each of the strand will form a new strand. Now here yellow strand has formed the green strand. So from one DNA, two copies have been produced. So further, this DNA will open up. This DNA can also open up like this. And this can lead to the formation of another strand like this. Similarly, this one strand, this will also form. So this formation of many copies of DNA, this is called as DNA replication. Like we will see what is DNA. This is DNA which is double stranded, and this part of the DNA is called as cistron, initiating codon, termination codon. Now this gene which is going to form RNA, this is called as cistron. Now we can see this strand, this part of the strand, supposing we are talking about this strand and this part, so they are going to form RNA, and this is called as template strand. This strand is called as template strand. So part of it will form the RNA. So whatever coding sequence is present on this yellow strand, which is called as template strand, now that coding sequence opposite to that, complementary in the messenger RNA, will be carried out. So we can say messenger RNA is carrying the messages present on the template strand of DNA. Like if we have A over here, in the messenger RNA we will have U. If we have C over here, here we will have G. So opposite sequence will be there, and this conversion of DNA to RNA is called as transcription. This is called as messenger RNA because this is carrying the messages. To learn about structure of messenger RNA, you can watch my video of messenger RNA. Again, link is shared in the description box. So messenger RNA with the help of ribosomal RNA, transfer RNA, this will form polypeptide chain. So polypeptide chain later on, this can form various kinds of protein. They can show one-dimensional structure, two-dimensional or three-dimensional primary, secondary, tertiary or quaternary structure. So whatever coded information is present in the DNA, now that has been converted in the form of a protein. So this transfer of information has occurred in the protein. Now we can see here in this slide DNA, like we discussed, a part is called as cistron. This can form messenger RNA and this process is called as transcription. This is called as transcription. Now this RNA, with the help of ribosomal RNA, transfer RNA, to learn about the structure of all the RNAs, you can again watch my video of RNA. So with the help of these three RNAs, this will form a polypeptide chain. This process is called as translation. Now this polypeptide chain can express either in the form of structural protein or proteinaceous biochemical or enzyme. Now, if we see many physiological activities inside the plant or animal like photosynthesis, respiration, digestion process, they are catalyzed by enzymes. So, enzymes are basically protein in nature, but all proteins cannot be enzymes. They help in catalyzing the reaction, they help in fasten the reaction. Now, some of the polypeptides, they may turn into hormones, they may turn into transport protein, they may turn into storage protein. So they are proteinaceous biochemical, and some of the protein, they are forming structure of cell wall, cytoplasm, nucleus. So if we talk about animal, collagen, actin, microtubules, so they are forming structure. So they are called as structural protein. So this polypeptide can express either in the form of structural protein, it can be in the form of proteinaceous biochemical, it can be in the form of enzymes. Now other kind of protein can be storage protein, transport protein, various kinds of proteins are there. So what is the mechanism of gene expression? Like we have discussed, this involved biochemical genetics. So formation of RNA over the template of DNA or gene, part of the DNA which is forming RNA is called as gene. So that is called as transcription, and that part of the DNA is also called as cistron. So coding information, or it is present in the form of triplet codon on the DNA. Now that information is passed to the messenger RNA. That's why it is called as messenger RNA. Now that coded information is again translated in the form of polypeptide chain. So this translation is done with the help of three kinds of RNAs. Now next we will discuss what is central dogma. Like we have seen, whatever information was present in the coded form in the DNA, now that will be transferred to the messenger RNA. This process is called as transcription. So whatever information or message has been transferred from DNA to messenger RNA, that will be converted into polypeptide chain, and this process is called as translation. So we can say there is unidirectional genetic flow of information from DNA to protein. So hereditary information or hereditary characters have been transferred with the help of messenger RNA to the protein. So this is called as unidirectional flow of genetic information from DNA to RNA and then to the protein. So this is also called as central dogma. This concept was given by scientist Crick in 1958. So one-way flow of information from DNA to RNA, RNA to protein, like we have already discussed. This arrow is showing DNA replication. Now here we can see sometime what happens from DNA, RNA is produced, RNA, protein is produced. Sometime from RNA, DNA can be produced. Then again from DNA, RNA is produced, then proteins are produced. So this is called as circular flow of information. This is also called as central dogma reverse. This concept was proposed by Tamin in 1963. This is mostly found in cancer and tumor viruses. Here RNA acts as template for the formation of the DNA because DNA synthesis doesn't occur. So DNA will undergo replication to form RNA and transcription to form RNA. Later take part in protein synthesis and vice versa. So we can see in the previous slide how this reverse transcription is taking place. Like see, now this is the RNA. Now, this sometimes this may be in the form of process gene. Process gene is when a split gene is there, exon, intron. We will remove the intron and only exon will join. So that is called as process gene. Now that may undergo reverse transcription. RNA will produce DNA. So DNA is double stranded, RNA is single stranded, and this conversion of DNA from the RNA or formation of DNA from the RNA is called as reverse transcription. And for this whole process, enzyme which is helping is reverse transcriptase. Now from this DNA further RNA will be produced. Again this will be called as transcription, and from RNA polypeptide chain and then protein. This process will remain same, formation of the protein, but this protein has been produced from the process gene or from the messenger RNA. Messenger RNA has been converted to DNA. This is called as reverse transcription. So reverse flow of information is there. Otherwise information was from, we have seen, from DNA to RNA. But here information is from RNA to DNA. So this is called as reverse flow of information, and this phenomena is called as reverse transcription. And this is also called as central dogma reverse, or we can say DNA. When this form RNA, this is called as transcription, but from RNA when DNA is produced, this is called as reverse transcription. As we know, DNA can form its copies by replication. Now RNA can form the protein with the help of translation process. So this is called as reverse flow of information, that is when RNA forms the DNA, and this is catalyzed with the help of reverse transcriptase, and this is also called as central dogma reverse. So this is all about gene expression. Thank you for watching my video. If you like my video, please like, share and subscribe.